invited speaker for today. You probably all know that the banquet takes place today. It's here in this venue, 5th floor. So we'll see you there at 7:30.
Okay, it's my pleasure to introduce the invited speaker for this morning, Joelle Pineau. Joelle is an associate professor and recently was named William Dawson Scholar in the School of Computer Science at McGill University. She's also the co-director of the Reasoning and Learning Lab at McGill. Joelle earned her PhD in robotics from Carnegie Mellon University, coincidentally, in 2004. Her thesis was Tractable Planning Under Uncertainty. The revisers were Jeff Gordon and Sebastian Trun. As part of the work, Joelle invented the PBVI algorithm for approximate PUMDP solving, which led to a whole host of point-based PUMDP solving techniques. And she also worked on the Pearl robot, which provided healthcare to people in assisted living facilities. She has continued this work in probabilistic planning and reasoning and machine learning with applications to both robotics and healthcare, in particular personalized care and treatment strategies for chronic diseases, which will be the subject of her invited talk today. Among a lot of the service that Joelle has provided to the community, notably is that she was program and general chairs of ICML. It's my pleasure to welcome Joelle Pineau.
Thanks, everyone. It's great to be here. I had already some wonderful interactions with some of you, and I guess I am here with a message today. A little bit of a call to arms for people to start looking more at problems in healthcare. And I think there are many, many rich problems out there for people with expertise in planning. My goal for today is to describe one of these problems where we've been working for the last few years. And at the end, also give you some ideas of some algorithms we've designed for these kinds of problems. Because yes, when you're doing important applied work, there is space for designing new algorithms and coming up with new techniques. So I'll give you one case study of that from work that has been done in my lab. And this work actually came from the very early days when I showed up at McGill. I was quite interested in learning about the place. And it turns out that a remarkably large part of McGill University is the Faculty of Medicine. And so wherever I would go, I would encounter these people in different various university functions, and they would tell me about their research. What do you work on? What do you work on? And invariably, they had these really interesting problems, many of which, when I heard about them, seemed to me like, this is kind of a POMDP problem. And I don't think it was just me seeing POMDP problems everywhere. I think the model is incredibly rich to model decision-making problems, sequential decision-making problems under uncertainty. And so as a result of some of these discussions, I found myself working on quite a few of these problems over the years. The one I want to talk to you about today is work we've done on developing a new line of treatment strategies for people with epilepsy. Some of you may know a little bit about epilepsy. There's about 1% of the world population that suffers from epilepsy. It can be a very debilitating disease. The seizures hit, in particular, are often very unpredictable in terms of when they're going to hit. And so people who suffer from epilepsy have a lot of difficulty leading normal lives. You don't know whether you should go out, go to the office, start driving your car, if at any point in time you can be hit by one of these seizures. The treatment for epilepsy has made remarkable progress in the last 100 years. Some of this work was done at McGill. Wilder Penfield was one of the developers, the first initiators of neurosurgery for epilepsy. For about 75% of people, actually medication does a very good job at reducing the incidence of seizures. Then there's a number of people for whom you can actually use resection surgery to take away the part of the brain which is responsible for the initiation of the seizure. That happens in cases where that initiation focus is very localized. More recently, a new line of therapy that's been developed is the use of neurostimulation for epilepsy. So in that case, there's actually an electrode placed in the brain that delivers electrical stimulation to the brain in an attempt to abort or completely cancel the incidence of seizures. And so my interest was really in looking at how that neurostimulation strategy is applied, and in particular, trying to figure out if through methods such as reinforcement learning, we could improve the effectiveness of this particular strategy. The use of neurostimulation is actually quite recent. Some of the earlier work didn't look at neurostimulation for deep brain stimulation. They were actually stimulating the vagus nerve. I don't know how much anatomy do you know. I don't know a whole lot, but I'm told the vagus nerve runs somewhere in the back of the neck. It's not really clear what is the mechanism through which stimulation of the vagus nerve would actually reduce the incidence of seizure, but it seems to work. Some people are trying to do deep brain stimulation. There's a trial that's underway, it's completed now, but was underway over the last few years, where they actually implanted electrodes in patients over several months. And after 25 months, in this case, they had about 90 patients in this study, 81 to be precise, that completed the 25 months of the study. And for a number of them, all the bars that are pointing down in that leftward graph, those are all people who had significant seizure reduction. 6 of these people went seizure-free after this procedure. Median seizure reduction was 56% in terms of the incidence of seizure. So there's definitely some early evidence that this can be a useful strategy for people with epilepsy. One thing that I should clarify is that both of these trials were done with what I would call open loop strategies. So it means that the parameters for the neurostimulation device are set by hand by the treating clinician. And there's a recording electrode in there that actually sees what's happening in the brain in real time, stores it in a device that the clinician can then look at. But that information of what's going on in the brain in real time, the EEG signal, is not used in real time to determine the parameters of the stimulation strategy. And so the work we started discussing with colleagues at the Montreal Neurological Institute involved looking at a closed loop version of this system, where we would actually in real time use the information from the EEG signal as essentially a state representation and use that to determine the choice of action that should be applied, therefore allowing us to modify the parameters of the stimulation strategy, whether it's the timing, the frequency, the intensity, in real time in response to the signal going on in the brain. And so this is really where I see this question of adaptive neurostimulation being phrased as a sequential decision-making problem. Unfortunately, we've been looking in my lab at sequential decision-making problems for a number of years. We have developed quite an expertise in doing that. All of our work was done in robotics, in simulation, and so on. So we had some catching up to do in terms of how are we going to take these methods that we've used in these robotic systems and apply them in these healthcare systems. And when we started digging more into this, it turned out that we were really in a major current here in terms of trying to do adaptive strategies for the treatment of chronic illness. So we're not the only group who's looking at phrasing these questions of sequential decision making for improving healthcare. You, I'm sure by now, have heard about the notion of personalized medicine. In most cases, personalized medicine is used to refer to the use of genetic typing of information to decide what type of patient subgroup you should be assigned to, and then your choice of treatment is determined based on this genetic typing. What we're doing here is a little bit different in that we don't want to just personalize the treatment to your patient subtype, but in fact we want to personalize the treatment over time in terms of how your disease is evolving, how you're responding to certain medication, how you're responding to different treatment strategies. And so again, we're not the only group doing this. There's been some very interesting work looking at adaptive treatment for alcohol addiction, for depression, for attention deficit disorder, and more recently, the folks that are working in oncology are very interested in these types of strategies, and we've been starting to have discussions with people doing both radiation therapy and chemotherapy for applying these same kinds of methods in this field. Of course, we can't necessarily do all of this work. There's a great need that I'm hearing, especially in recent years, from the medical professionals. So this is in that way that I'm encouraging you to go back to your universities, have these conversations with some of your colleagues in these areas, and you may find also that there's some really interesting problems to be solved in that direction. So let me tell you how we phrase this problem a little bit more specifically. We adopted the Markov decision process framework. Some of you may ask, why didn't I actually choose a POMDP over an MDP? And I'll tell you a little bit more about that in the longer term. But essentially, we didn't want to have to deal with the whole belief inference because we didn't feel we had a really good model of the system. And so to try to do belief tracking and so on requires usually having a good model of your dynamics. This is one of the really big challenge in this field, the fact that we don't know our probability transition function. We don't know our reward function. And so we really have to synchronously think of doing learning and planning at the same time. In terms of our objective function, we're looking, as in standard MDPs, at maximizing the expected sum of rewards over time. We don't usually look at a discount factor. We assume we have a finite horizon. We are learning a value function. This notion of a Q function will come up again a little bit over time in terms of how we're optimizing the strategy. So we've defined the Q function in the standard Bellman equation way, where we're maximizing the immediate reward plus the sum of subsequent reward. And essentially, we're just trying to pick the strategy that has the highest expected reward in terms of the reward function that we've defined. So just to recap, what's our objective? To bridge that gap from this MDP formalism with which you're very familiar to this application problem with which you're a little bit less familiar probably. The big goal, what we're aiming at, is to dynamically adapt our stimulation parameters. We considered a great number of different dimensions to that control action, whether we want to optimize where the stimulation was going to be applied. That turns out to be difficult given the hardware and the current state of neurosurgery. So we decided to pick the simulation site based on our clinical knowledge rather than optimize that. The other two parameters we started looking at were the frequency of the stimulation, right? At what timing should we be delivering this neurostimulation, the pulses, as well as the intensity of the signal. In the work we've done so far, we are mostly looking at optimizing frequency rather than intensity. They have a pretty good protocol for picking the intensity for each individual, and it seems that intensity can be held fixed through time rather than be modulated, whereas what's really important for epilepsy is really what's the timing of the pulses as a function of the neural activity in the brain. So in most of the work I'll describe subsequently, we're really just optimizing the timing of the pulses. We have two simultaneous goals, it turns out. Yes, we want to suppress seizures. We want to reduce the duration, reduce the incidence of seizures, but that's not the only thing we want to do. If that was the only thing we wanted to do, if we just wanted to achieve maximal suppression, there's very easy policies for doing that, right? Just drive up the intensity and frequency, and then you will achieve maximal suppression of epileptic seizure, as well as of all other brain functions. So that seemed less than optimal. So we looked at a dual objective where we want to maximize suppression, but we also introduced a criteria where we wanted to minimize the incidence of stimulation. In our case, that means minimizing the frequency. And we can just look at minimizing the number of actual pulses that are delivered to the brain. Last but not least, we really wanted to learn the strategy directly from the data. We didn't want to assume some very simplified in silico model of epilepsy. We wanted to have real data from the biological system to learn this. And we wanted to personalize the strategy to each individual. And so not necessarily have one policy that applies equally to all of the patients, but really that responds to the particular characteristics of that dynamic system. It seemed like a bit of an ambitious goal to try to carry out with human subjects. It was judged quite experimental by some of our colleagues, and so they suggested that maybe we should start by doing this in an animal model of epilepsy. Given that we wanted to learn directly from data, maybe do a little bit of exploration and then test out some policy, that also seemed like the most reasonable choice on our end. So we started with an in vitro model of epilepsy. So that means in this case that we are looking at animals, rats. We don't look at the live animals. We consider a slice of the rat brain. The slices that we look at include the hippocampus and the entorhinal cortex. These animals are not naturally epileptic. And so what we do is after that we have prepared the slice, we put it in a medium, in a dish, in a solution, And in that solution, we add a particular chemical called 4-AP, and that chemical induces what we call electroform behavior. So they're not really seizures in the sense that it's not a live animal, but if you look at the EEG signal, it looks remarkably like seizures do in live animals or in humans. And so this is a model that's been in the neuroscience literature for many years. They use this model to try to investigate the nature of epilepsy, the underlying mechanisms from a biological point of view. So we didn't really create this model. We just used this model of epilepsy to start asking the questions of optimal control, which we were interested in investigating. And in particular, in this case, for those of you who do know a little bit more brain anatomy, we're applying the stimulation directly to the subiculum, which is part of that slice model, and we're doing our recording in the entorhinal cortex. There's discussions in the literature about what's the best place for applying stimulation for this particular animal model as well as for humans, but that, from the clinical point of view, from our colleagues, seemed to be the best model for our purposes. So here's what a seizure looks like in this particular model of epilepsy. The middle part, that's the big deflection down to the bottom and the increased activity, that's what a seizure looks like in the EEG. We call that the ictal event. And before the ictal event, there's usually what we call a pre-ictal phase. That pre-ictal phase is characterized by these little spikes that are a semi-regular pattern. So if a person has suffered an epileptic seizure, when they actually, after that, go for clinical follow-up in the hospital, what the doctors are looking for, because most of the time the person won't necessarily have another seizure at that time point, what they're looking for in the EEG is these pre-ictal spikes. Most people who don't have epilepsy don't have these pre-ictal spikes in their brain signal. So that's very characteristic of a system with epilepsy, the pre-ictal spike preceding the seizure. So we have the seizure, the actual ictal event, and after the seizure there's something called the post-ictal phase. And that essentially means after the seizure. That's characterized usually by lower activity. And what happens in a seizure, essentially, if I give you sort of the cartoon version of it, is that all of the cells in your brain, all the neurons in your brain, actually start firing in a synchronous manner. They all get recruited, and all together they decide it would be a great party to all fire together at the same time. And after they've all fired together at the same time, then they kind of go into hibernation. There's a little bit of a rest period. So that's what the post-ictal period is like. It's very, very unlikely that a new seizure is going to happen during that time. In fact, all the cells are in that, all the neurons, are in that refractory period where they essentially don't have the ability to fire because some of their chemicals have been depleted from too much partying. And so what we're really trying to do when we apply neurostimulation, at least that's the hypothesis of the mechanism for neurostimulation, is that by applying electrical stimulation, you're forcing some neurons in that area to fire, which are forcing other neurons around it to fire. So it's like creating mini block parties all over. And as a result, they can't all be recruited to have one big party later on. And so if you apply that neurostimulation once in a while, it kind of disrupts the ability of the neurons to synchronize and cause a seizure. What I'm showing here in this slide is a periodic neurostimulation policy. So in this case, the stimulation has been applied at fixed time points without really considering what's going on in the brain. And in fact, there's good evidence that something as simple as a periodic strategy can be effective in disrupting seizures. What I'm showing you here is data from the lab with which we're collaborating. This is Massimo Avoli's lab at the Montreal Neurological, and they had already been doing these experiments before we started talking to them. These are some of the experiments that got me really interested in doing this collaboration. What you're seeing on the left is the top line is what happens when you don't apply any stimulation. So we call this a control condition, and you see a nice, beautiful seizure. It's downsampled from the seizure I showed you earlier, so it looks a little bit cleaner than the other one. After that, they looked at applying stimulation at 0.1 hertz. So that means every 10 seconds, there's a little electrical shock that's delivered. And in that case, you see that the seizure seems to be reduced in duration a little bit, but not necessarily significantly. They have a recovery period where they let the slice go back to standard level. Then they apply 2 hertz stimulation, 0.5 hertz, and 1 hertz stimulation. What we see when you get to 1 hertz stimulation, so you deliver a pulse every second, is that the seizure is effectively gone. And so they already had published some results showing that with 1 hertz stimulation, you could achieve significant suppression on this particular slice model. And so our job, in a sense, was to see if we could achieve suppression that was as good as 1 hertz, but with fewer stimulation pulses. In terms of effectiveness, there wasn't a whole lot of room to improve because they were getting quite good effectiveness with this particular model, but they were using quite a bit of stimulation. So could we reduce the amount of stimulation? Could we get equal effectiveness? And could we do so without programming in that 1 hertz was the right stimulation frequency? So in terms of MDP representation, because we were using the prior data I just showed you to train our model, our action space was set to be one of these fixed frequency policies, but we were going to allow the MDP to choose how to mix these strategies at every second. In terms of state features, we were looking at the EEG signal. In general, you can have multi-dimensional EEGs. In this case, we were looking at just one dimension of the EEG to make things a little bit simpler. We were looking at 10 to 15 seconds of EEG recording. The exact length is picked such that we have like a base 2 number of samples so we can run a fast FFT. So I think it was about 13 seconds that gave us kind of the right number. Over that, we extract Fourier transform features, so all the coefficients of your Fourier transform get put into one big feature vector, and we include range and energy information from that signal, and we end up with the feature vectors with about 80 different features in there. Our reward function had to include both of our components, so we had, in fact, we considered a cost function in this case, and we had a particular cost whenever a seizure was observed over that 1 second window, and we had another cost for when stimulation was applied over that window, and the weight between these two costs was actually controlled by a parameter alpha. So I'll show you some results where we vary this alpha, where we can get more stimulation or less stimulation in our policy based on how we set that. But for now, we manually set that alpha. And in our training data, which was obtained directly from, this is the data that we use for training, the data from this particular experiment. So in our training data, what we had to do is ask some of the electrophysiologists to look at the data and actually manually annotate where the seizures had been observed so that we can actually associate the cost function with that for the training data. Once we had the definition of our MDP representation, we looked in the literature, very rich literature for solving MDPs in the case where you're doing that directly from data. And there was a class of methods in there called fitted Q iterations that I think are very nice methods for doing batch reinforcement learning. And in the insight of these fitted Q methods is essentially that you're treating the problem of learning this Q function as a regression problem. So you take all of your samples from your training data, you cut them up in N tuples where you have state, action, reward, next state, call that an experience. Take all of these experiences as training samples for your Q function, and you're trying to learn, in fact, the Bellman equation through these samples. And so you can fit your Q function using a variety of different function approximators. We looked in particular at using random forest for doing that based on work that Damien Ernst and his team had done, showing really good experimental results for this particular framework, but you could use your favorite function approximator. One of the main advantages of random forest from our point of view is that there are very few parameters, hyperparameters to tweak, so that makes it quite easy to apply. And there's also good consistency properties, so you get more data, you can get better performance, and so on. So I'm not going to take you to the pain of how we trained all of this out. We had some software to do that. And this is, in fact, that kind of policy that comes out of doing that. So what you're seeing here in the top line is the EEG trace, and what you're seeing in the bottom line is the policy that we applied. So each of these red little line corresponds to a stimulation action that was applied. In fact, if you look deep down in it, each of the line is a square pulse that lasts 100 microseconds. That's usually how the stimulation strategy is applied, even if you're doing it periodically. And so if you look at the strategy a little bit, it's hard to analyze a policy, it always is. But there's two things that seem to characterize the behaviors that we see. One of them is that in the pre-ictal phase, where we're seeing the little spikes, there tends to be more complex policy decisions that are happening. The system is really trying to mix some of the higher frequency strategies in an attempt to desynchronize the system. And after we've successfully aborted the seizure, in that what would be the postictal phase, in fact, the system realizes that the synchronization has been broken, and what we see is a nearly periodic strategy, but the periodicity of that is actually lower than the 1 hertz, closer to 0.2, 0.4 hertz in that second phase of it. Now, this is all very qualitative. One of the hypotheses that one of the students in my lab had, Keith Bush, was a postdoc in our group, and came and really thought that we could see in some of the earlier phases the system essentially querying the dynamic system, so using stimulation to try to test out how excitable is the system, and that had evolved automatically out of the policies we were learning. Now, that would be a typical POMDP behavior, right? POMDP have these information gathering actions, and it seems here, because we're folding in 13 seconds of data into our state representation, we can effectively learn a policy that depends on some history. And it seems like there's some indication that our policy is using stimulation not just to abort the seizures, but really as a testing mechanism to see, how excitable are you, and if you turn out to be excitable, then I'm going to stimulate you more to make sure that I desynchronize the activity. Now, we haven't been able to confirm that hypothesis in a really convincing way yet, but that's something that seems to be very interesting arising out of these types of observation. So we had a strategy. It seemed to do interesting things. Interesting isn't quite good enough to get some publications in the neuroscience field. So we needed some real experimental validation, and so this is the comparison that we ran. First, we were going to test out periodic pacing at 1.0 hertz, because that's the standard right now for this particular model in the literature. Second, we were going to test out, of course, our adaptive stimulation strategy, something like the policy I just showed you. And third, we were going to consider another strategy. Now, this one's a little bit interesting. We wanted to see whether it was what our system was learning is the right periodicity for this particular system, or whether there was some advantage to be gained from having a non-periodic strategy. So the third strategy that we're doing effectively is set to deliver the same number of pulses as the adaptive stimulation strategy. So we always apply it afterwards. For a particular system, we look at over your 20-minute period of adaptive strategy, how many pulses did you deliver? Now let's take the same number of pulses and place them equally in that third strategy. So we have periodic pacing at what we call the effective frequency, which is calculated specifically for each slice after we've done condition number 2. And this is the result we got. So what we see in this case is that in terms of on our x-axis, we have the incidence of seizures. In terms of reducing seizures, both the 1 hertz and the adaptive are very, very good. The adaptive seems maybe slightly better, but really there's no statistical significance between 1 hertz and adaptive in terms of the efficacy. And what we're seeing is that the effective frequency is actually less good than the other two, though it's definitely significant over the control condition. So you can achieve some suppression with effective frequency, but not as good as with 1 hertz or with the adaptive strategy. And at the top, what we're showing is what was the effective strategy for all the slices that we observed. And what we see is that with this box plot is that on average, the effective frequency was more around 0.7 hertz, so significantly less than the 1 hertz frequency that is the current standard. Now I'll draw your attention to one fact here. If you look up top, what we have is n equals 11. That's 11 slices. That's not a huge amount. It's enough to have statistical significance. The reason I draw your attention to it is maybe as a bit of a word of caution, in that running these types of experiments is incredibly challenging compared to running experiments in stimulation, and even compared to doing robotics work. For these 11 experiments, it took us about 18 months, right, from the point where we had the optimal strategy and we just wanted to do the validation in the animal system. So that didn't get us discouraged. After 18 months, we still wanted to see what we could do to transfer these results. They were very promising on this animal model, but let's be realistic. The type of seizures we get on this animal model, the slice model, is very different from the type of seizures that we get in live animals or in humans. And so now we're really in discussions with clinical partners to try to see whether we can do this with real animals. One of the big challenges is that the types of seizures we get vary a lot. In the slides, the seizures look the same from one seizure to the other. They all look very, very similar. In the animal models, they vary a lot. So from one seizure to the next in the same animal, they can be very different. From one animal to the other, they can be very different. So there's a lot of work to be done in terms of that. It's also not clear what's the best open loop strategy for these live animals. So in the slice model, there were already published results saying when her strategy works quite well. In the live animals, we don't have these kinds of results. Of course, we need to deal with higher dimensional state and action spaces. And so these are all very interesting challenges. It's not all clinical challenges. In those 18 months where we were waiting for these 11 slices to be tested, we did a lot of other work. And we identified a number of computational challenges that we had to deal with in moving to the next phase. A big one is how are we going to acquire the data that we needed, right? For the slice, we used the data from the fixed experiment, the fixed strategies. In the live animals, there weren't such data available. We actually had the opportunity to carry out exploration, but with that opportunity came some responsibility not to use too many animals for unnecessary purposes, so try to design some efficient data collection strategy, have enough data that we could find a good policy, figure out how we were going to acquire the reward signals. When you have more data, it becomes more difficult to ask the electrophysiologist to manually annotate the seizures. Sometimes they disagree about, is this a seizure, is this not a seizure? So some questions to be resolved there. Of course, we needed to look at how we were going to use function approximation for estimating the return. The regression trees are great in terms of their performance. We were very happy with them. They don't always are the best in terms of scaling to very, very large data sets. So in this case, yes, we only have a few animals, maybe 10 animals, but the data we get from them is over a very long period of time, and it gets cut up in these 1 second segment. So it turns into many, many hundreds of thousands, tens of thousands in terms of the number of samples that we need to learn the policy. So we had to think a little bit carefully about this question. And finally, because we were eventually hoping to convince people to use this strategy for treating individuals, we had to think very carefully about what kind of guarantees could we make about the quality of the policy that we were going to deliver. It's not really good enough to just say, well, I have a policy which on average seems to be doing better than this other policy. We needed to have some real notions of how we were going to express confidence over our policies. So we did work across all of these problems. I'm not going to tell you about all of this work. I've picked one piece of work to tell you a little bit more about in the remaining time, and that is some work we've done in terms of designing new algorithms for function approximation with very large state and action spaces. And so the work we did actually follows from previous work that had been done in reinforcement learning. I don't know how many of you are familiar with this work. I think it's a really, really nice piece of work on reinforcement learning called Kernel-based Reinforcement Learning, done by Ormoneit Sen in 2002. And I remember Dirk talking about this work in some NIPS around 2002, 2003, and thinking this is a really cute algorithm. And it's very simple. The idea is that the data you have is of the same type I've described earlier. So you've taken your trajectories and you cut them up in these little snippet of experience, and each snippet looks at one state, one action, one reward, and the next state. So in this case, the gray nodes are the states, and the light blue nodes are actually the next state associated with each of these transitions. So the idea of kernel-based reinforcement learning is the following. Given that you've been given a batch of these gray-blue pairs, that's your training data. Now you want to make a prediction at a new state. We're going to call it red S. That new state you don't know anything about. You don't have data specifically in that location. Assume here that you're in a continuous state space, and so you're not going to have covered your whole space. Each sample is in a different state. And so in this new state, red S, you want to know what to do in this new state. You haven't seen it before. And so the notion of what to do in this state, red S, is going to come from a kernel function. And so the kernel defines how close is red S to those other gray S you've seen before. You can think of using a Gaussian as your kernel function, though other types are possible, but we typically use Gaussian kernels. You have a Gaussian kernel around each of your gray S, And now you make your prediction in terms of transition probability, reward function, Q function at red S based on a weighted sum of what happens at the gray S, where the weight is given by that kernel function. So the prediction at red S is going to be closely associated with those gray S that are close to red S and very weakly associated to the other S. And if you do that, you can actually compute your Q function for red S based on this weighted K here is just that weight that comes out of your kernel function, weighted Bellman equation for the nearby states. So I've been interested in this kernel-based RL algorithm for a long time. I think it has very nice properties. I'll tell you more about the theoretical properties, but first maybe I'll mention that we had actually used this algorithm before to try to pick out good policies for individuals with treatment-resistant depression. So in some other work, I've put up the references, some of you want to read more about it, but previous work we had a data set of patients which were offered were treated under different medication, and we tried to look at how we could make prediction of what was the best treatment strategy, pharmacological treatment strategies for these individuals with depression. And so this is a bit of a complicated graph, but what we're showing is really your state space is their baseline depression symptom level. In the y-axis, I have another state feature. In that case, how much did they improve after a first line of treatment? And what I'm showing you here is the value function for subsequent treatments for these patients. So what's their expected outcome with these patients? So blue means not very good. So the top of the graph, top right of the graph is those patients that started with severe depression and didn't improve after one treatment. The outcome for those is not quite so positive. But in particular, in the top left of the graph, in the top bottom right of the graph, we had those patients that started out quite seriously, but then that improved quite a bit, and so we can do quite a bit better with them. And we can also extract the policy in this case and show the policy. And this is what I think is useful in terms of using this for clinical purpose, is that you get these relatively smooth policy in this particular case. So the different types of gray values here correspond to different treatment recommendations for the next stage of their treatment. So how could we enhance their treatment for the different people? And when you show this, present this to a clinical person, they can actually read the graph and understand what the policy is going to do. And maybe the last bit of information that's useful for interpreting this graph is you see in each of the little cells, in this case we discretize the state space, but in each of the cell I have a circle. Bigger circle means that my kernel has more support in that area. Small circle means my kernel doesn't have a lot of support. So in the corners, we just didn't have a lot of patients in those particular situations. And so we don't have, of course, anyone who starts with no symptoms, very low symptom, and has a huge improvement, right? That top left corner, if you already don't have symptom, you're not going to have a big improvement, right? So we don't have a lot of people. And so from these kinds of strategies, we get policies that are relatively interpretable, which is quite important for convincing our partners, our clinical partners, to use this. And you also get policies that are relatively smooth. And so it's not switching treatments into patients that seem very similar. And that's really a characteristic of using this kernel function over that. From the computer science point of view, one of the reasons this appeals to me is that the KBRL method has actually really good theoretical guarantees. There are not many other function approximation methods for reinforcement learning that guarantee a globally optimal solution. KBRL does under some mild conditions, and conditions have to do with the fact that you need to shrink the size of your kernel at an appropriate rate depending on the density of data that you have in a particular. So it's a little bit technical, and in reality, it's not that obvious how to do. But nonetheless, we tend to have good statistical properties for this. On the practical side, it's also nice because the performance always gets better when you put in more data. And as I mentioned, we have some very interpretable solution. So it worked out very well when we were looking at doing this for inferring treatments for depression. But now we were in this problem of trying to infer treatments for epilepsy. And in fact, in KBRL, to compute your kernel function, you need to do all the pairwise comparisons between the different points. And that usually has a cost that's quadratic in the number of data points that you have in your data set. That proved to be a little bit prohibitive for our epilepsy case, where we had tens of thousands of points. And so we had to think a little bit carefully about how we wanted to do this. Fortunately, right around that time, a new postdoc arrived in our lab, Andre Barreto, and he came with a trick in his bag. I love postdocs that arrive with a new trick that I don't know. So here was what he was proposing to use. It's called the stochastic factorization trick, and it applies when you have a stochastic matrix. So what's a stochastic matrix? Well, all of the entries here have to be greater than 0, and the entries along each row have to sum up to 1. That seems very familiar, right? That's what my transition matrices look like when you're dealing with MDPs. And Andre said, well, if you have a stochastic matrix, you can apply the stochastic factorization trick. We said, do tell. So the stochastic factorization trick, in fact, looks a lot like matrix factorization. And so the idea is that you can take a big matrix P and decompose it into the products of two matrices, D and K. And D is going to have the same number of rows, and K is going to have the same number of columns, but the inner dimension is going to be much smaller. So those of you who've looked at non-negative matrix factorization, this is the type of structure that you're used to, but in that case, you don't necessarily need to have a stochastic matrix. If you do have a stochastic matrix, there's one more interesting thing you can do, and that is you can actually reverse the order in which you multiply the matrices. So instead of doing D times K to get P, your transition probability matrix, you can do K times D and get a smaller matrix P. And that smaller matrix, P tilde, actually has many of the same properties as your original matrix, in particular in terms of stationary distribution. And so that turns out to be quite useful for solving the MDP, because we can show that if you solve this big matrix P, if instead of solving the big matrix P, you solve the MDP represented by that small matrix P tilde, you're effectively looking at a state aggregation case. And K and D tell you how the states in your original matrix P relate to the states in your compressed matrix P tilde. So there's been some work in the MDP literature about state aggregation, state abstraction, and so on. So this falls in that flavor, but these matrices K and D tell us how to get from the larger system to that more abstract system. I'm not telling you yet how to choose K and D. I'll talk a little bit about that later. It turns out to be somewhat of an open problem still. But if you have a good way to pick that smaller dimension, then you can use that stochastic factorization trick and then just spend all of your efforts solving that matrix P bar. So for now, let me tell you an easy way to pick that smaller dimension, which is what we've done for all the results I'm going to show. And that is, think of that small dimension as being a set of representative states. So think you have picked, in your case, a few representative states. Maybe you took all your data, run some clustering algorithm, pick the center of your clusters. These are your representative states. And so in that case, you can actually use the number of representative states to determine that inner dimension of K and D, the smaller dimension of K and D. Okay, so we had many interesting discussions with Andre and decided, great, we have KBRL, we have the stochastic factorization trick. Let's try to put these things together and see if we can achieve an algorithm that has similar properties as KBRL, but lower computational costs by exploiting the stochastic factorization trick. So here's a little bit how it looks. In this case, you have these new representative states. I'm calling them S green in this case. And so those are the states that are on the inner dimension of your matrix. And so you can think of your matrix K as representing the transition from S gray to S green, and the matrix D as representing the transition from the state S green to the next state S hat, S blue. And so effectively, by looking at K and D separately and learning this from your data, you have this new matrix D that gives you, using the kernel function, the distance from your original states in your sample to these inner states. They're shown by S bar over here. And then you have this matrix K that gives you the distance from these representative state S bar to the next state in your data set. We're keeping a separate matrix D and K for each action. You can learn a single DNK for all the actions. It tends to work a little bit better if you learn a separate DNK for each of the action. The reason being, remember, this represents your transition system. Most of the cases, each action are going to have a different set of transition probabilities, so that's why we usually set it up this way. You can apply the same thing to your reward function to also go from your full reward vector down to some compressed representation. And now we have our small matrix P bar that's the product of K and D. And we're going to solve the MDP described by P bar rather than the MDP represented by original system. Now that P bar is the smaller one, right? That's M by M instead of N by N. So we're still quadratic, but in something much smaller, essentially the number of representative states that we've picked. In terms of properties of this algorithm, for one, it's very simple to implement. It still retains the interpretable properties that KBRL had. The computational cost is still quadratic, but in this number of representative states, which we assume to be quite a bit smaller than N. In the worst case, it's N, but in many cases, we can deal with much, much smaller M. We can still show that we converge to the globally optimal solution. Of course, that requires a few more, lines of specification, like M eventually converges to N and so on. But we still have that good property. Empirically, the performance gets better when M tends towards N, right? Put in more and more representative states, performance is going to get better. When you get more data, as N goes to infinity, performance is also going to get better. Since then, we've worked out a few extensions, which I'm not going to present today in the interest of time. One of them is that we have a version of this algorithm that we can apply online. So in some cases, right, we're still linear in that number of points N. And in some cases that we've looked at, the number of points we need to learn the right policy actually gets too big. Andre had a nice version of the cart pole balance problem where he had actually 3 poles on it, all wiggling together, trying to keep them all up at the same time. The representation of that isn't so big. The number of dimensions in your state space isn't big, but the number of samples you need to learn that policy correctly is huge, in the hundreds of thousands. And then even storing in memory something that is linear in N proved to be infeasible. So with this online version, he shows that if you're doing updates to K and D with batches of data, what we call mini batch sometimes, right? Get 1,000 examples at a time and accumulate the updates to D and K, you can actually do it efficiently without ever having to be linear in N. We have another version where we can parallelize a lot of this, so these computations tend to divide up easily in a map-reduce kind of framework, so we can run a parallelized version of this to make things a little bit more efficient as well. And very, very recently, this last year, Andre has worked out a new algorithm for automatically picking the representative states that doesn't depend on clustering. It's an EM algorithm, expectation maximization algorithm, where we assume that the latent variables of the hidden system are actually those representative states, and he runs EM to do the inference over these latent variables. So if you're interested, there's a paper at ICAPS that deals with this particular case. Let me tell you how well it does when we're trying to tackle this for epilepsy. So we haven't done the case with the very, very large data set. We don't have that very large data set yet. So what we did is take the data that we did have and build a computational model, a data-driven computational model of epilepsy. For those of you who are interested in doing RL models, the model is available as part of the RL glue library, so you can try it out for yourself. And once we have a synthetic model like that, you can generate as much data as you want. And so we use that synthetic data to compare a few algorithms. What I'm showing you here, x-axis is the performance of a particular policy in terms of the number of stimulation action that's applied, and on the y-axis is the number of seizures that is obtained. So our real value function is sort of a mix of these two things depending on how you set your alpha parameter. But this gives you a sense that everything that's going to be closer to the bottom is definitely better, right? Less stimulation and less seizure. Hard to achieve, so we don't get a lot of solutions there. Everything that's going to be up in the top right corner is going to be worse. Lots of stimulation, lots of seizures. Definitely not really good policy. We're comparing the KBSF policy, the little blue lines, that's all this KBSF policy, kernel-based with stochastic factorization algorithm, which I've just described. That's how well it does. And the reason we have these 3 blue lines is because we're comparing 3 different cases in terms of trading off the number of stimulation versus the number of seizures. And so it has what we call sort of a very well-behaved performance in that, if you're penalizing stimulation more, you're getting less stimulation. If you're penalizing stimulation less, you're getting more stimulation, and so on. So the blue lines are KBSF. The red lines, they're all grouped at the top. There's 3 little red lines. This is using an LSPI solver. For those of you who are familiar, it's an alternative method for doing fitted Q-learning with a batch of data, which is quite commonly used. The advantage of the limitation of LSPI in this case is particularly glaring when you look at the right side of the figure, and that is on a log scale the number of seconds we needed to obtain that solution. So if I look at the top case, KBSF is fractions of a second. LSPI is on the order of 1,000, 5,000 seconds. In terms of quality of solution, I would argue that the KBSF solution are probably much better in that they're having much, much fewer seizures, albeit at the expense of a little bit more stimulation. And finally, in green, we have all the solutions if you're using the standard tree-based regression, which we had used in the earlier work with the in vitro model. So when I showed you the results of the slice that we had actually done on the animal that was using these random forest architecture. So in green, you have the results of the random forest. And again, you can tweak the parameters of the random forest differently, so that's why we're getting different types of performance. The real take-home message is that in terms of computation costs, though we love these algorithms in the small data set cases, they just don't scale nearly as well as KBSF for these types of problem. So let me wrap up quickly by mentioning maybe very, very briefly 2 other projects that we are involved in, for which I have absolutely no results, but I have a great deal of excitement in terms of the impact we can have. One of them is in the design of an artificial pancreas system. What's very nice in this case is that the hardware for doing this in humans is already there. We have closed-loop systems that have real-time monitoring of the blood sugar level, the ability to deliver insulin through an insulin pump in real time. Right now, most of these systems require a human in the loop to calculate the dosage of the insulin, and so there's really very, very nice potential for using reinforcement learning methods. And in particular, because the dynamics of that system are very hard to characterize. Your blood sugar level depends on many, many factors, including your metabolism, what you're eating that day, the level of your activity and exercise, and so on. So it's hard to use classical control methods for doing that. And there's a lot of interest in looking at reinforcement learning from some of our clinical partners. We're also talking to a few different groups of using similar types of models for improving the treatment of cancer, both in terms of radiation therapy, where in the radiation therapy case, usually the amount of radiation that is applied over a treatment, let's say a 6-week course of treatment, is always, similarly as in the epilepsy case that I delivered, the dose of radiation is assumed to be equal. We call that equal dose fractionation over the whole period. And there's good evidence that if you modulate the amount of radiotherapy that you're delivering, you can actually achieve better results. Maybe delivering more radiation early on to really try to kill some of the cancer cells, and then backing up a little bit to see how well the healthy cell can pick back up and so on. So use that modulated adaptive procedure to improve the quality of care. So let me just finish by sort of emphasizing this call to all of you who are interested in looking in challenging applications. There are many, many problems where instead of looking at some of the more standard robotics and simulation domains we've been looking at, we can apply methods very similar in healthcare strategies. And in many cases in doing so, there's some really interesting computational questions that arise, which you have an ability to target. I am here telling you about this work, which of course was done by a fantastic team. Andre Barreto and Donna Prekop were really essential in developing the algorithmic side of what I presented. And Keith, Bert, Arthur, Massimo, and Gabriella were really essential in developing the epilepsy side of things. So I'm going to wrap up here. Thank you very much.
Okay.
So considering this MPD abstraction wise the rest of the there's some that what I missed is, as you said, you factorize different actions separately.
Yeah.
So representative states, whatever they are, for action A and different representative states for action B. And also, the second question, representative source states can be different from representative target states, right?
Yeah, so we don't differentiate between these. We assume just a single set of representative states, and we use the same one for all of the actions, and we don't differentiate in terms of source and sink state. And there's good computational reasons for doing that, because then all of our matrices stay the same size, and then in terms of doing Bellman backups and all that, it makes things much simpler. We have a lot less overhead for doing that. It's possible you could get better performance in terms of accuracy by modulating your choice for different actions, but it's not clear either, given that these states are all interconnected, right? They're not at all IID samples. They're taken from trajectories. And so it's not clear, since you're reducing on state, you're doing state abstraction, it's not clear that you would get better results in terms of performance. It's certainly not clear you'd get better results in terms of computation, which is our real motivation here. It's possible that you can achieve something similar by picking a different kind of kernel function. So we pick some pretty simple kernel function here. We're assuming some smooth spaces. It's possible that there's a lot of work on the machine learning side, learning kernel functions and so on, that you can do better by adapting the kernel rather than adapting the choice of states. And there, in that case, not necessarily paying the computational cost. But very speculative at this stage, obviously. Fine. Yeah. In a sense, I think it's really about what information do you have. And there's some systems for which we know how to build a reasonable model, at least even just what functional form, what class of families to use. And there's other cases where we don't. Some of the work that I didn't talk about is our huge effort at building a computational model of this epilepsy data. And in fact, we sort of went both ways. I set one student on the data-driven way, which just builds a model that's able to generate synthetic data that looks like the real data, but that has no biological plausibility, and one student on a more mechanistic way that read all of the neuroscience literature about what's going on in epilepsy and tried to build really good model. Both achieved some measure of success, but I will say that the results I presented from the in silico model use the data-driven way. And so he is using basically work from building, just predictive models based on data. So look at the past data and try to predict the next data, that type of model. So we have used some model-based approach, but even there, I would call them really data-driven models rather than mechanistic model. The reason being that I think we don't have a good enough understanding of what the real mechanistic model should look like. In the case of radiation therapy, we did build a synthetic model that is biologically plausible, and we're using that. We use the data just to fit the parameters of that model. In that case, there were a few models in the literature that we could use. We tried it. It's not clear, though, what's going to be the ability to transfer from that synthetic model to the real system. My guess is that when we get to the real system, again, the data-driven model or the model-free approaches are going to do better, simply because there's too much of a gap still in terms of the quality of the models that we have.